Hey kids, how you boys doing?
It's no secret that obesity is a significant public health problem. For now, we have a list of drugs which are FDA approved for treatment of obesity. Among them, we can find semaglutide, better known as Ozempic. Semaglutide, together with liraglutide, belong to glucagon-like peptide-1 receptor agonist. This class of drugs was developed to treat diabetes type 2. And as we know, glucagon-like peptide-1 receptor agonists are the first-line drugs in patients with diabetes type 2 with established cardiovascular disease. The only alternative option in this category of patients is SGLT2 inhibitors. But also, nowadays, semaglutide is the first-line drug in treatment of obesity. And in terms of obesity, semaglutide is by far the most effective drug.
Is that a fact?
No, it's not a fact. It's just what I heard. It's just what I heard.
Who told you?
They?
The standard dosage of semaglutide is 2.4 milligrams subcutaneously. So, among FDA-approved drugs for treatment of obesity, there are two GLP-1 receptor agonists. It's semaglutide and liraglutide. Both these drugs injected subcutaneously, and they are considered the most effective drugs in treatment of obesity. So, first of all, we have to understand what makes these drugs so effective for weight loss. So, let's begin with the mechanism of action.
Listen, everybody. Be calm, cooperate, and this will all be over in a minute. Fuck down! Well done.
The major mechanism of action of GLP-1 receptor agonists is increasing insulin secretion. To explain this, we have to know the physiology of insulin secretion.
You know what I'm saying?
It's an interesting point. Come on, let's get into character.
Here we have pancreatic beta cell. Beta cells have GLUT1 transporter that provides transportation of glucose into the cell. Also, they have ATP-sensitive potassium channels, and we have to know that in rest state, potassium channels are open. So, because potassium is intracellular ion, which means that the concentration of potassium inside the cell is much higher than potassium concentration outside the cell. While potassium channels are opened, potassium constantly leaves cell. Also, beta cells have voltage-gated calcium channels, and in rest state, calcium channels are closed. So calcium, which is extracellular ion, in rest state cannot enter into the cell. Also, beta cells have intracellular vesicles that have already formed insulin and C-peptide. When glucose enters into the beta cell, glucose undergoes phosphorylation by glucokinase to glucose 6-phosphate. Glucose 6-phosphate undergo glycolysis with subsequent degradation in Krebs cycle and electron transport chain that results in production of ATP molecules. ATP molecules activate ATP-sensitive potassium channels. With activation, potassium channels close. If potassium channels close, potassium begins to accumulate inside the cell. With increasing positively charged potassium ions, membrane potential increase, for example from -70, membrane potential increases to -50. And the state when membrane potential becomes more positive, we call depolarization. And depolarization triggers activation of voltage-gated calcium channels. If calcium channels open, calcium by concentration gradient begin to enter into the cell, and increasing intracellular calcium triggers exocytosis of vesicle with release of insulin and C-peptide into the blood. The fate of C-peptide is rather prosaic. C-peptide comes into the kidneys, and kidneys excrete C-peptide from the organism into the urine. Insulin in the blood stimulates tissues to consume more glucose from the blood circulation, and because glucose begins to income into the tissues, glucose level in the blood decreases. In addition to this, beta cells have GLP-1 receptor, which provides regulation of insulin secretion. Glucagon-like peptide 1 in the blood binds to GLP-1 receptor. With binding, receptor becomes activated. Activation of GLP-1 receptor stimulates closing of potassium channels and opening of calcium channels. As a result, more calcium molecules accumulate inside the cell, and with increasing intracellular calcium level, exocytosis of insulin increases. Interesting that the amount of glucagon-like peptide in the blood is controlled by a specific enzyme called dipeptidyl peptidase 4. This enzyme provides inactivation of glucagon-like peptide. In simple words, DPP4 destroys GLP-1 molecules. If we prescribe GLP-1 receptor agonists, they provide stimulation of GLP-1 receptor. This potentiates the effect of GLP-1 molecules and cause increase in intracellular calcium. And the higher the amount of intracellular calcium, the more active becomes the exocytosis of insulin. So the release of insulin and C-peptide into the blood increases. Increase in concentration of insulin in the blood stimulates tissues to consume more glucose, and thereby this will cause decrease in blood glucose level. And this glucose lowering effect is exactly the reason why we use GLP-1 receptor agonists in treatment of diabetes.
Pretty smart.
Yeah, I got my moments.
The second effect is decrease in appetite. And to explain this, we have to recall the pathway of appetite regulation. The central structure that regulates appetite is arcuate nucleus. Arcuate nucleus has two major types of receptors. It's neuropeptide Y receptor, let's call it NPY receptor, and NPY receptor stimulates appetite, which leads to increase in food intake. Additionally, arcuate nucleus has mineralocorticoid receptor, let's call it MC receptor. The stimulation of mineralocorticoid receptor causes decrease in appetite, which leads to decrease in food intake. The function of arcuate nucleus is controlled by two groups of neurons. We can divide them based on their effect on appetite. Orexigenic neurons stimulate appetite, and anorexigenic neurons cause decrease in appetite. Anorexigenic neurons produce melanocyte-stimulating hormone, which stimulates MC receptor, and by this it decreases appetite. Orexigenic neurons produce two substances. The first one is neuropeptide Y. Neuropeptide Y has two mechanisms of action. The first one is that neuropeptide Y stimulates NPY receptor, which causes increase in appetite. And additionally, neuropeptide Y inhibits the activity of anorexigenic neurons. The second substance produced by orexigenic neurons is a Gaucher-related peptide. A Gaucher-related peptide inhibits MC receptor, which leads to stimulation of appetite. All these processes that we have discussed occur in the hypothalamus, but the function of orexigenic and anorexigenic neurons depend on peripheral signals, and they receive signals in form of small peptides produced by various tissues in our body. The first substance is ghrelin. Ghrelin is produced by the stomach, and the function of ghrelin is to stimulate the activity of orexigenic neurons, thereby increasing appetite. Ghrelin is the only substance which has truly orexigenic effect. Because of this, we call it hunger hormone. In addition to this, duodenum and to a lesser extent other GI tract regions produce cholecystokinin, peptide YY, glucagon-like peptide 1, and oxyntomodulin. All these substances inhibit the function of orexigenic neurons, which leads to a decrease in appetite. And the last two substances are leptin and insulin. Leptin is produced by adipose tissue, and insulin is produced by pancreas. Leptin reflects long-term energy storage, because the higher the amount of fat tissue, the higher the production of leptin, and to accumulate fat tissue takes a lot of time. In contrast to this, insulin reflects short-term energy storage. We consume food and in response to meal intake, organism produces insulin. But we can unite them because they have a similar mechanism of action in terms of appetite. Both leptin and insulin stimulates anorexigenic neurons and inhibit orexigenic neurons. So, if we prescribe semaglutide, semaglutide potentiates the effect of GLP-1. So GLP-1 potentiates inhibition of orexigenic neurons. So by potentiation of GLP-1, we potentiate inhibition of orexigenic neurons. With inhibition, orexigenic neurons decrease the production of neuropeptide Y, which results in less potent stimulation of NPY receptor. As a result, appetite stimulation decreases, which results in suppression of food intake. Additionally, decrease in neuropeptide Y secretion causes disinhibition of anorexigenic neurons, so their activity increase, and increase in activity of anorexigenic neurons cause increase in production of melanocyte-stimulating hormone. This leads to a more potent stimulation of mineralocorticoid receptor, and as a result, it potentiates appetite suppression, which leads to decrease in food intake. Also, inhibition of orexigenic neurons cause decrease in production of a Gaucher-related peptide, which in turn cause disinhibition of mineralocorticoid receptor. The more potent is the stimulation of mineralocorticoid receptor, the more potent is appetite suppression, which leads to decrease in food intake. So, as we see, GLP-1 through three mechanisms cause appetite suppression, which in turn cause decrease in food intake.
And?
And nothing. That's it.
It's by far the most important mechanism of Ozempic in terms of weight loss. The third Ozempic mechanism is based on inhibition of gastric emptying, which leads to decrease in food intake. Usually, when we consume food, this food comes into the stomach, where it causes pressure on gastric walls. On gastric walls located gastric mechanoreceptors, and the pressure applied by food on gastric walls cause stimulation of gastric mechanoreceptors, and activation of mechanoreceptors give us a sense of satiety, which logically leads to decrease in food intake. After hydrochloric acid partially digests food comes into the duodenum, and the passage of food from the stomach into the duodenum we call gastric emptying. If we prescribe GLP-1 receptor agonists as semaglutide, they inhibit gastric emptying, which means that more food will remain in the stomach, and with increase in food volume in the stomach, the stimulation of gastric mechanoreceptors increase. The more activated are gastric mechanoreceptors, the higher the sense of satiety, and thereby the less amount of food we will consume, and decrease in food consumption leads to weight loss.
Found it! I knew she was hoarding it. I knew it. Always lying, saying she's out. Bald-faced liar. Mike! I found the chocolate pudding!
Okay!
The fourth effect of GLP-1 receptor agonist is based on decreasing insulin resistance, which occurs mostly secondary to weight loss. To explain this, we have to recall the effect of fat tissue on insulin resistance.
I am on a curiosity voyage, and I need my paddles to travel. These books are my paddles.
5 at a time.
Are you shitting me?
Excuse me?
What the hell?
Mr. Anderson!
So here we have tissues. Let's take muscle tissue for example. And there is pancreas. And pancreas has a lot of beta cells that produce insulin. Insulin in the blood binds to insulin receptor on tissues. With binding, insulin activates insulin receptor substrate, which is a part of insulin pathway. Activation of insulin pathway eventually leads to activation of phosphatidylinositol 3 kinase, which determines the insulin effect on tissues. Why do tissues need insulin? Tissues have GLUT transporter, and in the blood we have a lot of glucose. And as we know, glucose is a fuel for tissues.
Vanilla wafers, apple, banana, and trail mix.
Seriously?
We need energy for our travels, for stamina.
So, GLUT transporter provides the transportation of glucose from the bloodstream into the tissues. But GLUT transporter by itself is very slow. Let's suppose that GLUT transporter provides uptake of 2 glucose molecules from the blood and transport them into the tissues. Insulin effect on tissues can markedly increase the speed of GLUT transporter. And with insulin stimulation, GLUT transporter can deliver additional 7 glucose molecules into the tissue. So, as we see, insulin effect determines the amount of glucose that comes to the tissues. And at the same time, insulin effect determines the amount of glucose in the bloodstream. In addition to this, we have to know that glucose level in the blood is also maintained by liver production of glucose and by kidneys reabsorption of glucose. And all these factors help us to maintain normal blood glucose level.
Do you understand what he's talking about?
No.
Guys, come on, just think about it.
But also, we have a substantial amount of adipose tissue in our organism. And adipose tissue can release into the blood pro-inflammatory cytokines as TNF, interleukin-6 or resistin. And also, adipose tissue can release a lot of free fatty acids. Tissues have TNF receptor and toll-like receptor 4. Pro-inflammatory cytokines and free fatty acids can bind to these receptors, and with binding, they can activate various pro-inflammatory pathways inside the cell. In addition to this, free fatty acids can diffuse into the tissues, where they undergo lipolysis with formation of diacylglycerol. And accumulation of diacylglycerol inside the tissue can activate nucleoprotein kinase C. Activation of nucleoprotein kinase C causes inhibition of insulin receptor sensitivity, and also it inhibits intracellular insulin pathway. And this inhibition of insulin effect provides counterbalance to insulin effect on tissues. So it also helps to maintain normal blood glucose level. The problems begin when the amount of fat tissue increases. In some people, the mass of adipose tissue becomes huge, and we call this state obesity. In obese individuals with increase in adipose tissue, the production of pro-inflammatory cytokines and free fatty acids increase. This causes increase in stimulation of pro-inflammatory pathways, and also this causes increase in diacylglycerols. Together, overactivation of pro-inflammatory pathways and accumulation of diacylglycerol cause overstimulation of protein kinase C. Overactivation of protein kinase C cause overinhibition of insulin receptor sensitivity and insulin intracellular pathway. Altogether, this causes significant decrease in insulin intracellular effect. With decrease in insulin effect, the insulin stimulation of GLUT transporter decreases. So now, GLUT transporter will transport 4 glucose molecules less. As a result, the amount of glucose molecules in the blood increases, but the amount of glucose molecules in the tissue decreases. So now, in the tissues will be 4 glucose molecules less, but in the blood there will be 4 extra glucose molecules. In response to 4 extra glucose molecules, pancreatic beta cells will increase insulin secretion. So, in response to insulin resistance, beta cells are trying to produce more insulin molecules to overcome insulin resistance by increasing total insulin concentration. And at this point, blood glucose level will be normal, simply because extra insulin helps to utilize extra glucose. And eventually, this will cause dysfunction of beta cells, because nobody can work at his limits all the time. So at some point, beta cells will be no longer capable to produce so much insulin. And without additional insulin molecules, organism won't be capable to overcome insulin resistance. As a result, more glucose molecules will remain in the blood, and persistent increase in blood glucose level due to the insulin resistance is basically diabetes type 2.
I don't feel good about this. I don't feel good about this!
You feel good about anything!
If we prescribe semaglutide causes decrease in appetite, which leads to decrease in total amount of fat tissue. With decrease in fat tissue, the production of pro-inflammatory cytokines and the release of fatty acids decrease. This causes decrease in activity of pro-inflammatory pathways, which subsequently leads to decrease in activity of protein kinase C. Decrease in activity of protein kinase C disinhibits insulin receptor, which immediately increases the sensitivity of tissues to insulin. In addition to this permits to disinhibit insulin pathway, which in turn leads to increase in insulin effect on cell. With increase in insulin effect, the transportation of glucose by GLUT transporters increase, which permits to transport more glucose molecules from the blood into the cell. So this can restore tissue glucose level, and at the same time it helps to lower blood glucose level. And with decrease in blood glucose level, the load on pancreatic beta cells decreases, which can prevent their dysfunction. So this increase in sensitivity of insulin receptor in tissues and potentiation of insulin effect in tissues we call decrease in insulin resistance, which is one of the semaglutide mechanisms involved in weight loss.
You didn't know that?
Seriously?
Am I done here?
Son of a bitch, you're really no help at all, you know that?
Hey, language!
And the last mechanism of action is increase in lipolysis, which could decrease hepatic steatosis and lipid content inside the liver. Mostly, this effect occurs secondary to weight loss, but according to new data, this can be explained by alterations in oxidative phosphorylation. Studies shown that intake of liraglutide cause formation of smaller lipid droplets and cause increase in expression of genes related to lipolysis. This effect can be explained by stimulation of oxidative phosphorylation, by increase in expression of uncoupling protein 1, which cause browning of adipocytes. The same browning of adipose tissue was observed with intake of semaglutide. Increase in oxidative phosphorylation cause increase in oxygen consumption, and additionally, GLP-1 agonists cause increase in expression of hormone-sensitive lipase. So let's explain how all these findings are connected and how they cause weight loss. So it's mitochondrial outer membrane, its inner membrane, and its intramembrane space. On inner mitochondrial membrane located electron transport chain that is composed of 4 protein complexes and coenzyme Q. And also there is 5th complex called ATP synthase that has its own unique function. The general concept here is that protein complexes in electron transport chain by transportation of electrons suck energy out of electron. 50% of energy complexes use to drive hydrogen protons from mitochondrial matrix to intramembrane space, but another 50% of energy goes to generate heat, to maintain our body temperature. This hydrogen movement occurs at the level of complexes 1, 3, and 4. The major idea of this hydrogen transport is to lower protein concentration inside the mitochondrial matrix and to increase hydrogen protein concentration in intramembrane space. This process results in creation of hydrogen protein concentration gradient through the inner mitochondrial membrane. With high hydrogen concentration in intramembrane space and lower concentration in mitochondrial matrix. And ATP synthase uses this concentration gradient, and by transportation of hydrogen proteins by concentration gradient into the mitochondrial matrix, it generates energy. This energy, generated by ATP synthase, we use for phosphorylation of ADP, that results in production of ATP molecules. So, protein complexes suck energy out of electrons one after another until electrons will be completely energy depleted. And after complex 4 sucks the last portion of energy out of electron, now electron has no energy inside. Such electron is a wasted material that doesn't have any value for electron transport chain. So complex 4 throw out this low energy electron from electron transport chain to oxygen inside the mitochondrial matrix. And electrons bind to oxygen and reduce oxygen with formation of water. But how electrons are delivered to electron transport chain? For this purpose, we have mobile electron transporters as NADH and FADH, and each of these transporters has its own site of delivery. NADH transport electrons to complex 1, and FADH transport electrons to complex 2. NADH is by far the most important transporter.
What?
And most of NADH molecules we generate by degradation of acetyl-CoA in the Krebs cycle. In turn, most of acetyl-CoA molecules we generate from fatty acids during their beta oxidation, and fatty acids we get from triacylglycerols, which are basically lipid droplets. Semaglutide causes increase in expression of uncoupling protein 1 on inner mitochondrial membrane.
Fuck me, hold tight. What's that?
It's me belt, Turkish.
No, Tommy, there's a gun in your trousers. What is a gun doing in your trousers?
It's for protection.
Protection from what? The Germans?
Uncoupling protein 1 is also known as termogenin. This protein creates a leakage site for hydrogen ions that can bypass ATP synthase. The difference is that termogenin, in contrast to ATP synthase, do not generate energy. So termogenin causes decrease in hydrogen protein concentration gradient. But how it helps to lose weight? The logic is that the lower is hydrogen concentration gradient, the lower is the production of energy by ATP synthase per 1 electron, and decrease in energy production causes decrease in ATP production. But without ATP, our cells cannot live. So the only option for them to maintain energy production is to increase the delivery of NADH to electron transport chain. Basically, they try to compensate decrease in quality by increase in quantity.
What's wrong with this one?
Oh, nothing, Tommy. It's tip-top. It's just I'm not sure about the colour.
And where we get NADH molecules? The most obvious source is triacylglycerols. Basically, it's our fat tissue. So here comes the second effect of semaglutide, which is increase in expression of hormone-sensitive lipase. With increase in activity of hormone-sensitive lipase, the release of fatty acids increase. This causes increase in acetyl-CoA, and thereby it causes increase in generation of NADH molecules. And most important is that generation of NADH molecules is proportional to the breakdown of fat tissue. So by this we can degrade fat tissue which is already present in the organism, and it can be the explanation of loss of visceral fat on Ozempic therapy.
Have I made myself clear, boys?
Yeah, that's perfectly clear, Mickey, yeah. Just give me 1 minute to confer with my colleague. Did you understand a single word of what he just said?
So, as we see, semaglutide can lower blood glucose level, reduce body weight, and incredibly that it can reduce visceral fat, which always was very difficult. It can lower blood pressure, improve lipoprotein balance, and reduce fatty liver. And in addition to this, it causes dose-dependent decrease in gastric emptying. How Ozempic is effective in practice?
I don't know. That's a good question.
And the answer is that it's the most effective agent for weight loss in our possession. In first clinical trial in which adults with obesity took Ozempic once weekly for 68 weeks, the weight loss on Ozempic was 15.3 kilograms compared to 2.6 kilograms on placebo. In another clinical trial where diabetic patients with obesity took semaglutide for 68 weeks but in different dosage, the weight loss on 2.4 milligrams was 9.6% from the baseline. In case of 1 milligram, it was 7%, and patients on placebo lose 3.42%. So, semaglutide has dose-dependent effect, and 2.4 milligrams is the most effective dosage.
Good. Great.
In the next clinical trial in which patients with obesity took 2.4 milligrams of semaglutide for 68 weeks, showed 16% decrease in body weight compared to 5.6% with placebo, which is almost 3 times higher. The next clinical trial showed that the most significant decrease in body weight occurs between 60 to 68 weeks. In the next clinical trial with obese patients, semaglutide caused 15% decrease in body weight compared to 2.6% decrease with placebo, so it was almost 6 times higher, and the weight loss peaked at week 60. Here we have a graph with weight loss dynamic, and as we see, maximal weight loss occurred between week 60 and 68, which is a pattern of semaglutide effect. In the next clinical trial, they compared different dosage of semaglutide in participants of Asian ethnicity, and results showed 13% decrease in body weight with 2.4 milligrams, 9.6% decrease with 1.7 milligrams, and 2.1% decrease in case of placebo. So we can say that semaglutide is a very potent agent for weight loss. And the last clinical trial showed that semaglutide is by far the most effective agent for weight loss in patients with diabetes type 2.
Oh, that's cool.
The most common side effects of Ozempic are gastrointestinal reactions, as loss of appetite, nausea, vomiting, diarrhea, and abdominal pain.
So that's it then. No one else really knows anything. Okay.
But there is also very significant cosmetic side effect, and it's Ozempic face. The problem is that Ozempic is so effective that it causes dramatic fat loss throughout the whole body, including face. Physiologically, volume makes our face look useful, and in some parts of the face, this volume is created mostly by fat tissue. And if we give to a patient semaglutide, the initial effect of semaglutide will be weight loss with decrease in fat volume. This will provoke skin sagging in areas as temples, cheeks, near the jawline, and in nasolabial folds. Subsequently, this can cause changes in size of the lips, cheeks, and skin, which will cause disbalance of facial features, so it's the second phase. And most notable changes occur in elderly population, simply because they have reduced amount of elastin and collagen, so their skin is more prone to sagging. Probably the best illustration will be facial fat grafting, but in reverse. As we see on second image, there is highlighted fat tissue, and on third image we can see how the volume which was added by fat tissue improved the overall appearance. But with rapid fat loss, the volume will decrease, and the most notable changes appear under the eyes, in nasolabial region, and in mandibular regions. As we see, fat loss causes skin sagging. In young age, it's not so critical, because hollow cheeks are popular, but in elderly patient it can cause premature aging.
What did we learn, Palmer?
I don't know, sir.
I don't fucking know either.